A maize trial field in mid-August. Stefan Hertel and his scientist colleagues from the University of Bayreuth have a lot to do. The decisive moment for their experiments with bees is when the stalks bloom, because only then are the bees able to gather maize pollen. On each of the 32 field lots there is a flight tent containing 2 bee colonies. The scientists want to find out if the pollen from genetically modified Bt maize harms the colonies.
Wir versuchen hier auf diesem Versuchsfeld zu quantifizieren.
We're trying here in this field to quantify how honeybee colonies develop when they collect pollen exclusively from one type of maize. This means that these insects don't have the chance to fly around freely, but to here in these tents and collect only the pollen that's available there. We look at what's called a brood index, which means that we follow a single egg over its whole development until the bee hatches, and with this information we figure out an index. We can use it to reach conclusions about how various colonies differ in their development.
Vielleicht in verschiedenen Versuchsgruppen unterscheiden in ihrer Entwicklung. Des Weiteren schätzen wir halt also diverse
In addition, we estimate a variety of colony parameters, which means that we look at mortality in the bee colonies, at the general brood development, at the amount of pollen brought in, and, ultimately, how many bees remain in the colony after the experiment as compared with before.
Vor und nach dem Versuch halt noch in den Kolonien vorhanden.
Since not all maize varieties are blooming yet, not all bees are in the fields. The bees used in the trials were bred here at the Institute for Bee Research in Celle. In order to be comparable with each other, they all have the same genetic background. At the Bee Institute, newly hatched bees are being marked today. The 1- and 2-day-old bees are hungry. They need large amounts of protein in this phase of life. Later in the field, they will have access exclusively to maize pollen as a protein source. And by being marked, they are easy to find and recognize in order to check how much pollen they have taken up. Also known as beehives, this is what the artificial nests look like.
Hier halt der Futterbehälter.
Here's the food container. It's blocked off from the rest of the room, that is, from the combs, and this is known as a queen excluder. The queen can't get through, since otherwise the bees would build combs in here too and the queen would lay eggs in them, and we don't want that. Here you can see a cell that's been extended. That's a prepared queen cell. Since maize doesn't produce nectar, the bees are given a supplementary sugar solution. In addition, in each of the tents in the trial fields, there is a trough with water. The scientists have a daily routine. First, the exact development stage of the maize is determined. Then it is checked if dead bees are in the catchment. Flying activity is observed, and bees that are found on the maize blossoms are counted. In order to check how the individual colonies are developing, the beehives are opened and the number of bees are estimated according to a specific method.
75.
In order to look inside the honeycomb, the bees are shaken off. A single egg can be recognized on the floor of each comb cell.
Hier läuft die Königin an die Seite.
The large bee marked with green is the queen.